Welcome to the course on Introduction to Complex Biological Systems. This is the last lecture of week 7. Today I am going to discuss photosynthetic carbohydrate synthesis. So, this is a summary slide that will show you what I am going to discuss today. We have already seen the light dependent reactions of photosynthesis, right. So, we have seen that light is used to generate these high energy molecules. So, ATP from ADP and NADPH from NADP plus. Right. So, these high energy molecules are synthesized. That is something we saw in the last lecture. Today, we are going to see that these high energy molecules will be used to fix carbon dioxide. Right. So, 3 molecules of carbon dioxide will be fixed to produce a 3 carbon molecule, which is called a triose phosphate. Okay. So, this triose phosphate will then be the source for all these other molecules that are synthesized using various biosynthetic pathways. So, for example, the triose phosphate will be converted to a hexose phosphate. So, 6 molecule sugar. That hexose phosphate can be converted to, so this will be fructose. It will be converted to sucrose, which will be transported to different parts of the plant. So, non-synthetic non-photosynthetic parts of the plant, they will use this sucrose as the sugar source. Hexose can also be stored as starch. So, this is for storage. It can be also used to synthesize cell wall components. It can go into the pentose phosphate pathway that we have already seen, which produces all these metabolic intermediates which are used to synthesize DNA, RNA, proteins and lipid. Right. Now, in animal cells, the synthesis of carbohydrates in animal cells it always employs precursors with at least 3 carbons. So, they start from here, right, and these 3 carbon molecule can come from the catabolism of glucose or any other carbohydrate source that we have consumed as food, right. However, plants can synthesize these carbohydrates and all of these things directly from carbon dioxide, right. So, plants can synthesize carbohydrates from carbon dioxide and water, and they use light as the energy source. So, in the last lecture we have seen chloroplast as the site of action. Now, I am going to introduce another term which is plastid, right. So, it turns out that all types of plastids are bound by a double membrane. So, we have seen that in case of chloroplast, we have seen that in case of mitochondria. So, plastids are that, and mature chloroplasts have extensive internal membranes. So, mature chloroplast is also a type of plastid. However, mature chloroplasts have this extensive internal membranes that we saw in the last lecture are thylakoids, right. The internal membranes can be lost, as you see here, when a mature chloroplast becomes a proplastid, which is this, right, and resynthesized. So, they can be resynthesized, right, but it goes not like this, but in this pathway where it goes to this pregranule plastid, right. So, a protoplastid gives rise to a pregranule plastid and then a mature chloroplast. Protoplastid in non-photosynthetic tissues such as root, they can give rise to amyloplast, which is here, which contains large quantities of starch. All plant cells have plastids, and these organelles are the site of important processes such as synthesis of essential amino acids and vitamins. So, all the essential amino acids and vitamins, they are synthesized in these amyloplasts. Okay. So, now let us look at the carbon fixation. Just like the citric acid cycle, this is another set of cyclic reactions, right. So, here we are going to see carbon fixation, right, and this cycle can be divided broadly into 3 stages. Stage 1, where fixation of atmospheric carbon is done. Stage 2, where reduction happens, and stage 3, where this acceptor molecule, which is ribulose 1,5 biphosphate, it is regenerated. So, this cycle is called Kelvin cycle, right. So, let us look at the number of carbons, because we have to keep track of the number of carbons here. So, it starts with 3 molecules of ribulose 1,5 biphosphate. Now, this molecule has 5 carbons, 1, 2, 3, 4 and 5, right. And there are 3 molecules, so 5 times 3 will be 15. So, we start with 15 carbons. 3 molecules of carbon dioxide come in. So, that is another 3 carbons. So, 15 plus 3, that is 18. Now, these two together are converted into 6 molecules of 3-phosphoglycerate. Now, 3-phosphoglycerate is a 3-carbon molecule, right. So, 6 molecules of this 3-carbon molecule. So, 6 times 3 is 18. So, we see that the number is conserved. 15 plus 3 was 18, and here we have also 18. Now, these 6 molecules of 3-phosphoglycerate are reduced. So, this acid group is reduced to an aldehyde group, right. So, that is reduction, and that reduction is carried out by NADPH, which itself is oxidized to NADP+. So, 3-phosphoglycerate becomes glyceraldehyde 3-phosphate. This is also a 3-carbon molecule, and we have 6 molecules of this. Now, at this stage, one molecule out of these 6 molecules goes out of this cycle, and this glyceraldehyde 3-phosphate can be used for energy production via glycolysis. We have seen that it is one of the intermediates in glycolysis, or it can be used for production of sugar via gluconeogenesis or starch synthesis, right. The remaining 5 molecules will go back into this cycle. So, 6 molecules of glyceraldehyde 3-phosphate, that is 18 carbons, one goes out. So, one molecule of glyceraldehyde 3-phosphate will have 3 carbons. So, you can see that these 3 carbons are typically balancing with these 3 carbons. Okay. So, I am not saying that these 3 carbons make this extra molecule, but you will see that these 3 carbons are balanced by these 3 carbons, right. So, you can sort of say that 3 carbon dioxide now go out of this cycle as one molecule of glyceraldehyde 3-phosphate. Now, you have left with 5. So, 5 molecules which are each of 3 carbons. So, 5 times 3 is 15. They are converted to 3 molecules of ribulose 1,5 biphosphate. So, this is 5 carbon molecule and 3 molecules. So, 5 times 3 15. So, this balance is also maintained, right. So, essentially the 6th molecule of glyceraldehyde 3-phosphate, the 6th molecule is the net product of photosynthesis, right. So, these carbons are fixed, 3 carbon are fixed, and they go out of the cycle, and the remaining carbons are in the Kelvin cycle. So, this can be used to make hexose sugar for fuel and building materials such as sucrose for transport to non-photosynthetic tissues or starch for storage. So, this one glyceraldehyde 3-phosphate that can be used for these purposes. Now, all the 13 enzymes which are involved in this pathway are in the chloroplast stroma. So, this reaction or this cycle takes place in the stroma of the chloroplast. So, let us look at stage 1, which is fixation of carbon dioxide into 3-phosphoglycerate. This reaction is carried out by this particular enzyme called ribulose 1,5 biphosphate, which is one of the substrates, and it is carboxylated. So, ribulose 1,5 biphosphate carboxylase. However, we will see in a few slides that this enzyme also performs another reaction which is called oxygenase, right. So, instead of fixing carbon dioxide, it will fix oxygenase. So, that is why there is a slash here. So, ribulose 1,5 biphosphate, either a carbon dioxide is added to it or an oxygen molecule is added to it. So, that is why carboxylase / oxygenase, and this long name of the enzyme is shortened as Rubisco, right. So, this enzyme is also called Rubisco. This enzyme has 8 large identical subunits. Each subunit is 53 kilodalton in size, and it has another 8 identical small subunits. Each subunit is 14 kilodalton in size. So, together it forms a very large protein complex. Interestingly, the large subunits are encoded in the DNA that is present in the chloroplast, so the chloroplast genome, whereas the small subunits are encoded in the nuclear genome, right. So, these two proteins are synthesized in 2 different parts, and then they come together and they are assembled. This is one example of an enzyme which has a very slow turnover. So, one molecule of Rubisco fixes only 3 carbon dioxide molecules per second at 25 degrees centigrade, right. But this is a very important reaction, right. So, it turns out that chloroplasts have a very high density of this molecule. In fact, it turns out that among all the protein molecules that are present in a chloroplast, almost 50% of all protein molecules will be Rubisco, because you need this in a very large quantity. This is similar to what we have seen in case of hemoglobin, right? So, major part of the red blood cell is composed of hemoglobin. Similarly here, almost 50% of all the proteins that is present in a chloroplast is rubisco. And since this is such an important reaction and it goes on in all different plants, rubisco makes up the most abundant enzyme on this planet. This is the active site of rubisco. So, there are two important features that you should see. One is this lysine residue. So, this is a lysine residue, you see the lysine side chain, and then it will end with an amine group, so NH3 plus. However, this amine group is modified by a carbamoyl group. Okay, so this is carbamoylated lysine, and there is a magnesium ion in the active site. So, this magnesium ion is coordinated by this carbamoyl end of the lysine, then this aspartic and glutamic acid side chains are there. The substrate is also there, this is the ribulose 1,5 biphosphate, and finally carbon dioxide is there, right. So, this is the active site and we will see how the reaction happens. So, this is the carbamoylated lysine, okay. So, that is the carbamoyl group, right. It is coordinating with the magnesium ion, and this is your ribulose 1,5 biphosphate. So, we can count the number of carbons 1, 2, 3, 4 and 5, right. Now, this proton, so you see a ketone group here. The first reaction is the enol formation. So, you see that enol formation happens. So, enol formation happens here. At this point, this histidine 294, it acts as a general base. So, it abstracts this proton and triggers this reaction. Coordination of the carbon dioxide by the magnesium ion makes it a very good site for nucleophilic attack. So, this nucleophilic attack happens, so it is triggered by this. Okay, you can see the arrows are moving. So, this protein is abstracted, negative charge goes here, these electrons attack this carbon. So, this nucleophilic attack happens, the carbon is now connected here, right. Now, a water molecule comes in and what it will do is it will cause the hydrolysis of this bond. So, if we count the number of carbons at this point, 1, 2, 3 on one side, 1, 2, 3 on the other side. So, it hydrolyzes this into 2 3 carbon molecules, right. So, this intermediate that is formed is called beta keto acid intermediate, and now the hydroxylation occurs at this position and this bond will break and this molecule is released. So, this molecule is the first molecule of 3-phosphoglycerate that is released from rubisco. So, this is 3-phosphoglycerate. So, this is carbon 1, carbon 2 and carbon 3, it is phosphorylated at that position. So, it is a 3-phosphoglycerate. Now, this rest part is still attached in the active site, right. So, a proton will be donated by this lysine to this carbon ion, and now it will be released as a second molecule of 3-phosphoglycerate, right. So, 5 carbon ribulose 1,5 biphosphate and 1 carbon dioxide, total 6 carbons, they are converted to 2 molecules of 3-phosphoglycerate, both have 3 carbons. So, 2 3 carbon molecules are released. That is the first step, which is the fixation of carbon dioxide is complete. Now, stage 2 is reduction of this 3-phosphoglycerate to glyceraldehyde 3-phosphate. So, you see this is the 3-phosphoglycerate. So, 1, 2, 3, 3-phosphoglycerate. The first step is it is another phosphate group is added at this position, right. So, it becomes a ester and then it is reduced by glyceraldehyde 3-phosphate dehydrogenase to glyceraldehyde 3-phosphate. So, this acid group is reduced to an aldehyde group. So, this becomes glyceraldehyde 3-phosphate. So, again 1, 2, 3, this is the third carbon. Okay. So, our second stage is complete. However, some more reactions happen and it is important to show them here. Glyceraldehyde 3-phosphate can be converted to dihydroxyacetone phosphate, right. So, again it is a 3 carbon molecule, only thing is that instead of aldehyde it becomes a ketone. So, this group is shifted. So, it is done by an isomerase, which is triose phosphate isomerase. Now, one molecule of glyceraldehyde 3-phosphate and one molecule of this dihydroxyacetone phosphate, they are condensed together by the enzyme aldolase to produce fructose 1,6 biphosphate. Fructose 1,6 biphosphate, one of the phosphate group will be removed by fructose 1,6 biphosphatase. So, the first phosphate group is removed. Okay. So, this phosphate group is removed and it becomes fructose 6 phosphate, this molecule. Now, fructose 6 phosphate can be used to synthesize starch, which will be storage form of the sugar molecules, right. Another thing that can happen is this dihydroxyacetone phosphate can be transported out into the cytoplasm by this antiporter. It gets converted to glyceraldehyde 3 phosphate, and then this glyceraldehyde 3 phosphate, which is the same molecule here, can go into the glycolytic pathway. So, it can produce ATP and then it can produce further ATP molecules, right. On the other hand, these two molecules can again be combined to form fructose 1,6 biphosphate like this, which gets converted to fructose 6 phosphate like this, and then it is converted to sucrose, which will be transported to other non-photosynthetic parts of the plants, and there it will be kept as or used as energy source, right. So, all of these things can happen. However, we are interested in the formation of glyceraldehyde 3 phosphate, that will be as one of the products of this stage, right. Now, just for comparison, we have seen some of these reactions before. So, I am showing you it here, right. This 1,3-biphosphoglycerate, this is 1,3-biphosphoglycerate. We have seen the formation of glyceraldehyde 3 phosphate from this, you see, that is gluconeogenesis, right. So, glyceraldehyde phosphate dehydrogenase, the same enzyme is present here. Then triose phosphate isomerase converts it into dihydroxyacetone phosphate. Aldolase combines these two to form fructose 1,6 biphosphate. Fructose 1,6 phosphatase converts it into fructose 6 phosphate, right. So, these from here to here we have already seen in case of gluconeogenesis. So, these are the same enzymes that you will see in different parts. Okay. So, we have glyceraldehyde 3 phosphate formation at this point. Now, one molecule, okay, so let us go back. So, 6 molecules of glyceraldehyde 3 phosphate are synthesized, 1 molecule goes out, remaining 5 will be used to regenerate 3 molecules of ribulose 1,5 biphosphate. So, this is what we are going to see now. Okay. So, 1 molecule goes out like this or this, right. So, we will be left with 5 molecules of glyceraldehyde 3 phosphate, and 5 molecules of glyceraldehyde 3 phosphate will be used to regenerate the ribulose 1,5 biphosphate. That is stage 3. So, 3 molecules of glyceraldehyde 3 phosphate and it is converted to dihydroxyacetone phosphate by this triose phosphate isomerase, right. So, let us say 1 molecule is converted to this, 1 molecule remains as glyceraldehyde. Now, these two are condensed by aldolase to fructose 1,6 biphosphate. We saw this in the previous slide. So, it is a 6 carbon molecule, it is converted to fructose 6 phosphate. We have also seen this in the last slide. So, again it is a 6 carbon molecule. Now, this 6 carbon molecule and another molecule of glyceraldehyde 3 phosphate. So, remember we have 5 in our hand, this is 1, this is 2, this is the third one. These two combine, so this 6 plus 3, 9, these two combine to form 2 sugar molecules. So, 6 plus 3, 9 becomes 4 plus 5, 9. So, 1 molecule of erythrose 4 phosphate and 1 molecule of xylulose 5 phosphate, right. Now, see, we have a 5 carbon molecule, we need a 5 carbon molecule because ribulose is a 5 carbon sugar. So, this is converted to ribulose 5 phosphate by ribulose 5 phosphate epimerase. Okay. So, xylulose gets converted to ribulose and then another phosphate group is added by this ATP to convert it to ribulose 1,5 biphosphate. So, we have synthesized 1 molecule of this, we need 3. Okay. Now, the 5 carbon molecule is used up here, this 4 carbon molecule combines with another molecule of dihydroxyacetone phosphate which comes from glyceraldehyde 3 phosphate, right. So 4 plus 3 is 7. These are condensed to form a 7 carbon molecule by the same enzyme aldolase. You see, it's the same enzyme. So it forms pseudoheptulose 1,7 biphosphate, right? Biphosphate, one of the phosphate groups goes out, so it becomes pseudoheptulose 7 phosphate. Okay, so it is removed by this enzyme heptulose, pseudoheptulose 1,7 biphosphatase. So we have a 7 carbon sugar. Another molecule of glyceraldehyde 3 phosphate comes in. So 7 plus 3 is 10, right? Now again the transketolase comes in, the same enzyme that we saw here. So it converts this 3 plus 7, 10 carbon into two 5 carbon sugars. So one is xylulose 5 phosphate, which is the same as this, and again it will be converted to ribulose 5 phosphate by the same enzyme ribulose 5 phosphate epimerase, and that is converted to ribulose 1,5 biphosphate. So from this path we get the second molecule of ribulose 1,5 biphosphate. Here we have ribose 5 phosphate. This is converted to ribulose. This is ribose, this is ribulose. Ribulose 5 phosphate by the enzyme ribose 5 phosphate isomerase. So now we have again ribulose 5 phosphate just like this. So ribulose 5 phosphate kinase will use ATP to add another phosphate group to produce the third molecule of ribulose 1,5 biphosphate. Okay. So two molecules of ribulose 1,5 biphosphate came from here and the third one is here, right. So we used 5 molecules of glyceraldehyde 3 phosphate to produce 3 molecules of ribulose 1,5 biphosphate. And where are the 5 molecules of glyceraldehyde 3 phosphate? You can count them here. So this is the first molecule. This is the second one because this comes from the same glyceraldehyde 3 phosphate. Third, fourth and fifth. So these are the 5 molecules of glyceraldehyde 3 phosphate that produce 1 from here, 2 from here and 3 from here. 3 molecules of ribulose 1,5 biphosphate, which is this. Okay. So let's look at some of these steps little more carefully so that we can look at the actual structures of these molecules. So if we look at this step 3, fructose 6 phosphate and glyceraldehyde 3 phosphate, right. So this is fructose 6 phosphate, 1, 2, 3, 4, 5, 6 carbons, and this is glyceraldehyde 3 phosphate. What transketolase does is it will take these 2 carbons and put here. So now we have a 4 carbon sugar erythrose and a 5 carbon sugar xylulose. So erythrose and xylulose are formed and then it will go to 2 different paths. Erythrose combines with this to form aldolase, which is a 7 carbon molecule, and then that 7 carbon molecule combines with another 3 carbon to form 2 5 carbon molecules. So this is again catalyzed by transketolase. So let's look at that. So this is the 7 carbon molecule 1, 2, 3, 4, 5, 6, 7 and 3 carbon from the glyceraldehyde 3 phosphate 1, 2, 3. So 7 plus 3 is 10. Now it forms 2 5 carbon molecules ribose 1, 2, 3, 4, 5 and xylulose 1, 2, 3, 4, 5, right. And then these two go into these 2 pathways. So xylulose will get converted to ribulose and finally ribulose 1,5 biphosphate. Ribose also gets converted to ribulose and finally ribulose 1,5 biphosphate. So let's look at the energy usage for the production of glyceraldehyde 3 phosphate from 3 carbon dioxide molecules. So remember in Kelvin cycle 3 carbon dioxide molecules produces 1 glyceraldehyde 3 phosphate, right. So for every 3 carbon dioxide molecules fixed, 1 molecule of glyceraldehyde 3 phosphate is produced. And to do that 6 plus 3, 9 molecules of ATP and 6 molecules of NADPH are used, right. So very high energy is used to produce this 1 molecule of glyceraldehyde 3 phosphate. Now, if you remember from the previous lectures in this week, we saw that NADPH and NADH, they are similar. The only difference is a phosphate group on the sugar molecule, right? So, if I assume NADPH as equivalent to NADH, and we also saw that NADH is equivalent to 2.5 molecules of ATP, right? So, 6 molecules of NADPH will be equivalent to 6 times 2.5, that is 15 molecules of ATP. So, 15 here and 9 here, that is 24 molecules of ATP, that is the equivalence of ATP, are used to produce 1 molecule of glyceraldehyde 3-phosphate from 3 carbon dioxide molecules, right. So, 24 equivalents of ATP are used. If we think of production of glucose from this, then 2 molecules of glyceraldehyde 3-phosphate will be needed to produce 1 glucose molecule, right. So, in essence 24 times 2, 48 molecules of ATP will be consumed to produce 1 glucose molecule from 6 carbon dioxide molecules. Okay. So, we are using up 48 equivalents of ATP. Now, if you remember the complete oxidation of glucose via glycolysis, the citric acid cycle and the oxidative phosphorylation, it produces 32 molecules of ATP, right. So, here we are consuming 48 molecules of ATP to produce glucose, and when we oxidize glucose back to carbon dioxide and water, we produce 32 molecules of ATP. So, 32 out of 48 is 2/3 or 66%. So, we get back 66% of the energy that is used to produce glucose, right, which is not bad. Okay, that is very good efficiency, that we can actually get back 66% of the energy that has been used to produce these high energy molecules like glucose. Okay, and ultimately all of this energy is coming from the sun. So, solar energy is the source. Okay. Now, let us look at this other part that Rubisco does. So, it fixes carbon dioxide, that is what we have seen, but it can also fix oxygen to this molecule ribulose 1,5 biphosphate. Okay, and that process results in something which is called photorespiration. So, photorespiration is a process in plants where the enzyme Rubisco binds to oxygen instead of carbon dioxide, leading to a wasteful pathway that reduces the efficiency of photosynthesis, right. So, it turns out that in every 3 or 4 turnovers, Rubisco catalyzes the condensation of oxygen with ribulose 1,5 biphosphate. So, instead of carbon dioxide, it uses oxygen to form 3-phosphoglycerate and 2-phosphoglycolate. Okay. So, remember that when ribulose 1,5 biphosphate, which has 5 carbons, is fixed with carbon dioxide, totally 6, it will produce 2 molecules of 3-phosphoglycerate. But instead of carbon dioxide, if it is oxygen, we do not have this extra carbon. We have only 5 carbons from the ribulose 5-phosphate. So, 3 of them produce 3-phosphoglycerate and the remaining 2 produce 2-phosphoglycolate, which is a 2-carbon molecule. Okay. And this 2-carbon molecule is metabolically useless product. So, it has to be reconverted back to 3-phosphoglycerate. Okay. And that uses a lot of energy. So, this is a wasteful expenditure of the energy that was tapped by these light reactions during photosynthesis. So, this fixing of oxygen is the oxygenase activity of the enzyme. That is why this enzyme is called a carboxylase/oxygenase. Okay. The Km or the Michaelis constant for carbon dioxide of Rubisco is around about 9 micromolar, whereas that of oxygen is 350 micromolar. So, it binds carbon dioxide more tightly than oxygen. However, if you think about the substrate concentration, we have more oxygen than carbon dioxide. So, atmosphere has 20% oxygen and around 0.04% carbon dioxide. And if you think about the dissolved gas, then the concentration of oxygen is 250 micromolar, whereas that of carbon dioxide is 11 micromolar. So, if you have high substrate concentration, then of course, the saturation of the enzyme will also be high. So, since we have higher concentration of oxygen, even though its Michaelis constant is less, we will still get a higher saturation almost equivalent to that of carbon dioxide. So, that is why in every 3 or 4 turnovers. One is oxygenase reaction that is catalyzed by Rubisco, and this results in significant oxygen fixation and waste of energy. So, this is the reaction ribulose 1,5 biphosphate, right. So, this is the enol form that we saw. Now, oxygen binds here instead of carbon dioxide, and then it produces these two molecules. So, this is the good product 3-phosphoglycerate which can be used, but this is the useless metabolite which is 2-phosphoglycolate. Okay. So, this has to be somehow salvaged, and the salvage pathway is called the glycolate pathway. So, this salvage pathway of glycolate is costly. It converts 2 molecules of 2-phosphoglycolate to 1 molecule of serine and 1 molecule of carbon dioxide. So, 2 molecules of phosphoglycolate means 4 carbons, serine has 3 carbon, and the rest or the remaining carbon goes out as carbon dioxide, okay, which will again be captured by the Rubisco to do its fixation. Now, the hydrogen peroxide that is produced, so we will see hydrogen peroxide is produced here. It is neutralized by peroxidase enzymes in the peroxisome. Okay. So, let us look at the reaction. Let us say Rubisco takes up oxygen instead of carbon dioxide. It fixes it with ribulose 1,5 biphosphate to produce 1 molecule of 2-phosphoglycolate, which is not the useful molecule. So, this 2-phosphoglycolate, the phosphate group is removed, okay, by phosphatase to produce glycolate. Okay. This is CH2OH and CO minus. So, this molecule now goes out from the chloroplast into peroxisome. Here the glycolate is oxidized to glyoxylate using oxygen. Okay. So, glycolic acid oxidase is the enzyme which will produce this glyoxylate. So, the alcohol group is oxidized to an aldehyde group. Okay. Now, transamination reaction happens which adds this NH2 group here and it converts it into glycine. We have already seen this amino acid, right. So, CH2, NH3 and CO minus. So, this is glycine. This glycine now passes out from the peroxisome, goes into mitochondria. So, you see that it is passing through all these different organelles, right. 2 molecules of glycine are taken and glycine decarboxylase. This is a enzyme complex. So, there are 3 enzymes and this enzyme complex takes up 2 molecules of glycine, removes ammonia, releases 1 carbon dioxide and converts it into serine. So, you see there are 2 carbons. So, 2 glycine means 4 carbons. Those 4 carbons are converted to serine. So, you have 1, 2, 3 carbons and the remaining carbon goes out as carbon dioxide. Okay. So, that is why this is called a photorespiration. What happens in respiration? We consume oxygen and release carbon dioxide. So, here oxygen is consumed and carbon dioxide is released. So, this is equivalent to a respiration, and to distinguish it from the normal respiration or cellular respiration that happens in mitochondria, this is called photorespiration. Okay. Now, the serine molecule is passed back to the peroxisome where the amine group is taken off. So, deamination happens, right. So, this amine goes from here to the glycine and it forms hydroxypyruvate. Okay. And this hydroxypyruvate is now reduced to glycerate by this enzyme alpha hydroxy acid reductase. So, it uses NADH as the reducing agent which gets converted to NAD plus, and this glycerate goes in to the chloroplast. It is phosphorylated to produce 3-phosphoglycerate, right. So, this 2-phosphoglycolate finally produces this 3-phosphoglycerate. Now, 2 molecules of these are taken to produce 1 molecule of 3-phosphoglycerate. 3 carbons are here and 1 carbon is released as carbon dioxide, right. So, the combined effect of Rubisco oxygenase and the glycolate salvage pathway consumes oxygen, which is here, and produces carbon dioxide, which is here. This results in the name photorespiration. This inefficiency has led to evolutionary adaptations in the carbon assimilation process, particularly in plants that have evolved in warm climates, right? Because in warm climates, again, the binding of carbon dioxide becomes weaker. So, the photorespiration becomes even more dominant. So, let's look at the other pathways, right? So, one is carbon assimilation in C4 plants. So, plants that fix carbon to form 3-phosphoglycerate, as we have seen till now, these plants are called C3 plants. They are producing 3 carbon molecule. There are other plants where the formation of this 3-phosphoglycerate is preceded by several steps, one of which is temporary fixation of carbon dioxide into a 4-carbon compound, right? So, they produce a 4-carbon compound, and these type of plants are called C4 plants. Photosynthesis in the leaves of C4 plants involves 2 types of cells, mesophyll, which is shown here in green. So, this is mesophyll, and the other one is bundle sheath cells, which is shown here in blue, right? So, here what happens is phosphoenol pyruvate. So, this phosphoenol pyruvate in the mesophyll cells, right? They combine with bicarbonate ion. So, carbon dioxide is dissolved in water to produce this bicarbonate ion, and phosphoenol pyruvate carboxylase, this has very high affinity for this bicarbonate ion. Okay, so it does not have any affinity or much affinity for oxygen. So, here a filter works. So, phosphoenol pyruvate is converted into oxaloacetate by this enzyme phosphoenol pyruvate, right? Now, oxaloacetate is converted to malate, which passes on into the bundle sheath cells. Here it is oxidized to pyruvate, and 1 molecule of carbon dioxide goes out. This pyruvate goes back into the mesophyll cell. Phosphorylation of this happens by this enzyme pyruvate phosphate dikinase, and it produces this phosphoenol pyruvate. So, it regenerates this phosphoenol pyruvate. So, the net effect is that carbon dioxide is dissolved as bicarbonate, which goes into this cycle and is released here. And this carbon dioxide is taken up by Rubisco to produce 3-phosphoglycerate. Now, this whole pathway eliminates oxygen from this. So, Rubisco is not coming in contact with oxygen and hence works at its maximal efficiency, right. So, this reduces photorespiration in C4 plants. There is another pathway which is called the CAM pathway. So, succulent plants such as cactus and pineapple, which are native to hot and dry environment, they temporarily separate carbon dioxide capture and Rubisco action. Okay. So, the carbon dioxide capture that happens and its fixation by Rubisco, they are separated in time. So, how they do that? So, during the day when it is very hot, these plants reduce water vapor loss by closing the stroma. So, if you close the stroma, then of course, no exchange of this gaseous carbon dioxide or oxygen is happening. So, this prevents the entry of carbon dioxide also. No water is released, but no carbon dioxide is also taken up. However, at night when the temperature is low and hence loss of water vapor will be less, the stroma open. This allows entry of carbon dioxide. Carbon dioxide is fixed into oxaloacetate, which is reduced to malate as we saw in the previous slide, and then this malate is stored in vacuoles. Okay. So, it is immediately not used for. So, the malate is produced and it is immediately not used to release the carbon dioxide. So, this malate is stored in special organelles called vacuoles. Why store it in vacuoles? Because malate, it is malic acid, so it will release protons and it will reduce the pH, and this reduction of pH can damage other enzymes which are present in the cytosol or the plastid, right. So, to protect those enzymes, this malate is stored in vacuoles. During the day, carbon dioxide is released from the malate as we saw here. So, from the malate, carbon dioxide is released. This is happening in the day when the stroma is closed. So, no carbon dioxide is entering and no oxygen is also entering, right. So, this is released from the malate, assimilated by the action of Rubisco and Kelvin cycle enzymes, right. So, by this temporal separation of carbon dioxide capture into malate and release of carbon dioxide to Rubisco when the stroma is closed, so no oxygen is coming in, it reduces photorespiration. This process was first discovered in stone crops such as the family of flowering plants called Crassulaceae. Okay. So, that is why based on this, these are called CAM plants, and this pathway is called the CAM pathway. Okay. So, the last part that we will see is the biosynthesis of starch. So, during active photosynthesis in the day, a plant leaf produces more triose phosphate, which is glyceraldehyde 3-phosphate, than it requires for generating energy or producing other biosynthetic pathway materials, right. So, the excess molecules are converted to sucrose and transported to other parts of the plant to be stored or be used as fuel, and the another part is stored as starch. So, starch is the main storage form. So, in plants like sugar beet and sugarcane, sucrose is the primary storage form, right. So, starch and sucrose are synthesized in different cellular compartments. Sucrose is produced mostly in cytosol and starch is produced in plastids. So, this is something that we have seen earlier. So, I can go back to that slide and show you this one. So, sucrose is produced in the cytosol and starch is produced in the plastid. So, this is an example of a starch molecule. So, this is amylose. Okay. So, it is a linear chain of sugar molecules, and typically when you study sugar chemistry, you will see that this end is referred to as the reducing end of the sugar and this end is called the non-reducing end. I am just introducing these two terms because this will be useful in the next slide where we see the synthesis of this linear sugar molecule. So, the synthesis of this sugar molecule is done by this enzyme starch synthase, and it turns out that it has two sites where reaction happens, and the sugar molecule is activated as ADP glucose. So, this is your glucose 6 carbon sugar, and to it ADP is added, which is a very good leaving group, right. So, each of the two reactive groups XA and XB at the site of the starch synthase makes a nucleophilic attack on the ADP glucose. So, it displaces the ADP, forming a covalent linkage attachment to the C1 of the glucose subunit. So, this attaches to the glucose subunit, to the C1, C4. These are the two sites we are going to look at. So, both are attached to the C1, right. Now, the bond holding the glucose residue 1 to XA, this bond undergoes nucleophilic attack by the OH at C4 of the glucose residue on 2. So, this is the C4, right. So, this attacks here and this will attack to the next sugar molecule. So, what happens then is 2 is attached here after 1 and this 1 shifts here. So, XB to 1, this is what we get. So, 1 is shifted from here to here and in position of 1, 3 comes in, right. Now, the reverse attack happens. So, this will attack here. So, 2, 1 will get shifted here, see 3, 2, 1 and 4 comes here, right. So, it will keep on oscillating like this and the chain will grow on these two sites. So, here it is in site B, gets shifted to site A, then again gets shifted to site B, right. So, many repetition of this sequence extend the oligosaccharide adding glucose residues at its reducing end. So, this is the reducing end, it will keep on adding more and more sugars at this end, okay, and it can go on. So, depending on how many reactions happen, you will get a long polysaccharide. Okay. So, whatever I have discussed in this lecture, you can find in any biochemistry book, and again Leninger principle of biochemistry, you can use this as a primary source for studying this. Thank you.